Okay, so let's start today's class. So till this, we have learned about the basic concepts. Okay, so what we have studied, the reactivity, acidity. Okay, so these are the basic steps that we already have gone, and we have done some reactions. Okay, so addition, oxidation, as well as reduction reaction of alkenes. So today, what we are going to do, so we'll go through the brief recap of the substitution and elimination reactions. Okay, and the controlling factors. So which lead us to two different things. One is the stereo chemistry as well as chemistry. Okay, so let us start. So first we'll go through the substitution. By the name, you can understand that substitution reaction is such a reaction where a part of molecule, or especially it is termed as leaving group, so that is uh, that leaves actually, and then one more coming part that is called a nucleophile, it substitutes that leaving group. So this can be depending on the mechanism, this can be divided into two subparts or two types. One is called SN1 or unimolecular. Why it is called unimolecular? Because in the transition state, in transition state, one species is involved. Is involved, and the other one, which is called SN2, that is called bimolecular, because in transition state, two species are involved. So we'll briefly study about both uh, the substitution mechanisms, and what are the comparison, and also the idea. So conditions, molecules to paper SN1 or SN2, and then energy profile Well diagram, because this will give you an idea so that you can relate the energetics with the reaction. That is very important to understand. Now we'll start by the unimolecular one, question one. So how it goes? Suppose you have a molecule like this, so methyl, the tertiary group, which is connected to a leading group. Suppose this is bromo. This we call as leading group. What happens when you have some very weak nucleophile? So this is delta positive, this is delta negative. Okay, when you have very weak nucleophile, so what will happen? This bromo will leave at first, and then you will have what is called carbocation. Yes, Br minus. In the second step, there will be weak nucleophile, what is referred to as H2O. This one will come and will attach to the carbocation center. So what you will get? Then O, H, OH2, and this will be positive, right? So you cannot have this one stabilize, so it has to go through something. So another H2 will come and this will act as a base. Then it will balance this charge, and so that you will have your final product, that is this will be a product and this is the substrate. So one thing you can see that when this is a planar molecule, okay, this is a planar molecule. So this water, it can coordinate from this side, or there is another possibility that it will come and attack from this side. So you can have both the products. You will have like this. This can be formed, and if it attacks from the other side, then you will have the another word. So you can see that what is the relation between these two, A and B. There is a mirror plane out there. Mirror plane out there. So these are called enantiomas. Hmm? If one is R, then another one is S. Right? So what you will get in SN1 enantiomeric mixture will be the product. So what it implies that it is SN1 is not is not PVA specific. Okay, it is not stereo specific. So it will not give you particular one enantiomer. Okay, suppose you have something R or S anything, so you are going to get both the enantiomers in the end. And now let us go to the energy profile. Energetics of SN1. To draw the reaction coordinate, how it will look like. This is your energy, and this is the reaction coordinate. So you are starting with a reactant. Where are you going? You are going to a carbocation step, right? This is your carbocation. This is your TS1. Then what you are forming from here? You are forming another intermediate, which is this is another one, and then you are forming your product. You can see here, this will be the activation energy for your reaction. Okay, and that is the RDS. RDS is also called as rate determining step, which is the slowest step of the reaction. So depending on this, your reaction rate will be uh, based on the species that are involved in the RTS. You can see from here, that's RTS is rate determining step. Okay. We have to check the number of species involved in RTS to get the rate equation. What you can see here, rate is proportional to only one species is involved here. So this is it is depending on only one species. That means and also this is the substrate. So this will be the rate equation for SN1. You can see only one species, that is substrate, is involved. And another thing what you can understand from here, so how you will control the rate of the reaction. Okay, so this is your first intermediate, that is the carbocation. If you can make it somehow more stable, let's say here, you may have some different pathway, and then you will get the product. Okay. Carbocation has to be more stable in order to control reaction rate. Obviously, the faster. So you can see that in SN1 you have only substrate that you can vary. If you vary any other constant, like if you vary the nucleophile or the solvent, something like that, so this has very less effect on the reaction rate because reaction rate directly depends on only the substrate concentration. Okay, so this is one thing. And then if you can make this carbocation stable, so the energy gap, this energy gap changes. Okay, it reduces. So when energy gap reduces, much faster that intermediate is going to be achieved in terms of energy, and subsequently the rate will be the faster. Okay, so with that, you can see that more stable the carbocation, more faster is the SN1 reaction, right? And then you can write it as the benzylic, then allylic. These are the carbocations, okay, that tends to go through SN1 mechanism, okay, and this tends to go through SN2, will come later. But these are the type of carbon centers that will go through SN1 because these carbocations are much more stable. So, okay, carbocation intermediate. So for the same thing, this 1 degree methyl alkyl halide, halides do not undergo SN1. No stable carbocation intermediate. Okay, and in this scenario, leaving loop is an important component. Okay, why? So depending on your the leaving possibility, you have the differentiation of the good and bad living groups. So I will note it here. What are the good living groups you have? Cl minus, Br minus, I minus, H2O, F minus. Okay, then you have this acetate one, then you have OH minus, OCH3, and H2. So these are called root leaving groups while we are going to the pure leaving group. Okay, so generally it is seen that these are to consider the conjugate acid of this species, HCl, HBr, H I, hydronium ion, HF, acetic acid, water, methanol, and ammonia. So consider the pKs. So these are all less than 0. This is 3, this is 5, this is 16, 16, 55. So you have a differentiation based on the pKa value. So when it is less than 0, it is an intermediate, less than 10 and above 10. So these are called good, these are moderate, these are weak, depending on the pKa value. So you can see that when they are leaving, they are actually stable. Okay, they can stabilize the negative charge, whereas the other one they cannot. So leaving group remains a important part, and also have to talk about the nucleophile. What we know about nucleophiles, they are electron rich, looks for electrophilic or electron deficient species. Say, for example, you have this is R 2 bromo butane, and you react it with water. It's little bit of it, you will get to butanol. Okay. So how it will go? First thing is water is weak, so it will leave at first, and you will get this carbocation. Okay. Now H2O is it can come from here, from here, both sides, and then you can have both these intermediate, or you can have this one. The relation between them are enantiomers. Okay. Although expected is 50-50 mixture, but generally, mental inversion is more than retention. What is inversion? What is retention? That is the chiral center. Okay, or chiral center. That means R2S or H2R. Okay. With that, we move to the other one, that is SN2, bimolecular. What happens here? You don't have any intermediate state. So let us go through it. Suppose you have primary alkyl halide. So primary alkyl halide is methyl bromide. Then one nucleophile is there, and it will attack from the back side your leaving loop leaves. Then you come, there is no intermediate, so there is one transition state which much similar to like this. See, transition state you cannot isolate, you cannot get an experimental evidence, but you can visualize it. In this transition state, bromine is delta negative, which is also delta negative, and this is delta positive. So at the same time, bromine is going to leave and OH is coming from the backside, so that you will have the, the inversion of configuration. Okay. So what is the energy profile? This is very important. This is your energy axis. This is your reaction coordinate. Now this is your starting point, H3Br, and we are going to the product, that is H3H. This is your transition state. So what you can see here, you don't have, this is the delta G. So you don't have any intermediate, or there is only one transition. So how the rate, with what the rate will depend? Rate will depend on both your substrate and the nucleophile. Why? Because in this transition state you can see both these two molecules are involved. Okay, this is what it looks like. Uh, some constant into substrate and the nucleophile concentration. This will be the right equation for SN2. You can see the analogy is you take 3, 2 and 1 degree, then you have methyl 1. This has the tendency to go towards SN2, and with more complexity in the structure it will go towards SN1, more bulkiness if it's there. Like to compare between this ethyl bromide, propyl bromide, bromide with an isopropyl group here, and you have a tert butyl group at the end. So you can see that this carbon adjacent to or alpha to the boron of bromide is the same, but the bulkiness is changing here. There is this only one, two isopropyl tert butyl. So bulkiness is getting increased. So that SN2 reaction, SN2 there will be 1, 10 to the power minus 1, 10 to the power minus 3, and 10 to the power minus 5 order. So for SN2, you have a less sterically congested carbocation center. For the same reason, this vinyl groups or any living group attached to a double bond doesn't undergo SN2 mechanism. Okay, and you need a strong nucleophile needed for SN2. And regarding the stereochemistry, what we have seen that it goes mostly through the inversion. Okay, so let us take an example. This is stereochemistry. The living group is iodide here, and H to react with acid, which is nucleophile to iodobutyl. What you get here is the inversion of configuration. Is to iodobutyl. Okay, and here we will have only one product. That is the inversion, 100%. Okay, so one more type of SN2 is there, that is called intramolecular SN2. In this case, uh, in some particular conformation, you have both presence of nucleophilic and electrophilic center in a same compound. It can go through uh, intramolecular substitution. Okay, and there is also role of solvents. Role of solvents. Two types of solvents are there, which is called polar aprotic and polar protic. Right? Polar protic means water, any kind of alcohol, okay, then acid, and polar aprotic means anything that is not protic but polar, like the acetone, etc. So if you see this reaction rate comparison, and in polar solvents, yeah, much faster. So why does this happen? This is the solution effect of the polar protic solvent. So if you have something like that, they tend to go each bonding with nucleophile, okay, as well as the substrate. Okay, so either way it can work. It can sometimes increase the reactivity of nucleophile or it can decrease it also. Okay, so just for a comparison, on SN1 and SN2. So first thing is the case of SN1. We'll first try to see a general case. In SN1 you have starting, then one intermediate, and maybe one intermediate or not will come to the product. This is the carbocation. Now this is the case for polar aprotic case, where they have no stabilization of the extra stabilization of the carbocation. You have some solvent that can stabilize it, stabilize this carbocation, polar protic solvent. Then what you can see, your energy difference is less compared to this one. Okay, so reaction is faster. So one is faster. Compared to this, if you go to SN2, energy and reaction coordinate will have reactant product. It is going like this. This is polar aprotic. Now, if polar protic is there, then what will be? See, first polar protic with its bonding with substrate plus nucleophile. Here, as a result, nucleophilicity philicity decrease. So, slower reaction. So, nucleophilicity decrease, so slower reaction. So, this will be look like the energy difference will be more. SN2 is lower in protic solvent. Okay. So with that, we complete the substitution reaction cases, and now we move to elimination. Elimination reactions can also be same thing, E1 and E2, and this is competitive to substitution. How? Let's say you have bromocyclohexane and you are giving some nucleophile. So what it can do, it can replace this bromo one. You get the OR plus bromine. There is one another scenario where this OR will give you beta elimination. How? So you have the H, okay, on the beta carbon. This is alpha carbon, this is beta carbon. So this beta carbon, say this one, can be extracted by this nucleophile. Then this electron density will come here and the leaving group will leave. We will have this cyclohexane elimination product. And we will have ROH plus here minus. Okay. So these are some reduced selectivity. Like bromo, then methyl, methyl, and you have also this methylene group here. So if you do elimination, you can end up with either of these products, either this one or this. Generally, this one is thermodynamically stable. Okay, because this is much more substituted. Thermodynamically stable. So this product is formed more. Okay. So as we told, this has also E1 and E2. First we will start with the E1. Unimolecular. For this one also, it has to go through two steps. There is one intermediate, and the stability of carbocation matters. CH3, CH3, Br. This will be at first. So you will have the carbocation. Positive minus Br minus. This is your intermediate. Okay. And in the second step, what happens? Suppose I can write it like this: CH2 and H. Now, if you have a base here, then this photon can be abstracted. This electron density will fall on this bond, and you will have H3, CH3, and H2. This is your product, elimination product, and you will have BH. Okay. Let us go to the energy profile diagram. We reaction coordinate. You are starting from reactant 1 intermediate. This is your TS1. This is your product, and this is TS2. This is the energy required. Hmm. So here it depends on again the substrate. Okay, for E1. What is the case for E2? So from the name only you would have understood that this will be similar as SM2. Living group, alpha, beta hydrogen. Then you have nucleophile. Nucleophile will abstract the hydrogen. Directly it will come and replace it. So that you will have your product. In this case also you don't have any intermediate. So you have one transition state. Transition state will look like this. Important thing is that this proton which is being abstracted, it should be exactly 180-degree dihedral angle of the leaving group. Okay. Similar as SN2, the proton getting abstracted should be 180 degree, should be at 180-degree dihedral angle from the leaving group. What is dihedral angle? So suppose you have two planes like two pages of a book. Okay, you have two planes, and between these two planes you have some angle theta is called dihedral angle. Very important thing. Okay. Suppose you have, for this case, you have something like this. So this is 90, this is also 90. So altogether you have 180-degree dihedral between this one and this one. So from that, if we see the energy profile, what it will look like. You have product and very similar single arm curve, the reactant, and we are going to the product. So there is only one transition set. There is no intermediate. So here your rate will depend on substrate plus base or nucleophile, whatever. So both are in the transition step. So now important point is that stereochemistry in this kind of reaction is controlled by conformations. What is that? Let's say you have this nucleophile, exactly the thing I was telling before. We have E and one group here, D. These two are at 180-degree dihedral. Okay, so now if it attacks here, this will come and this will go, so that we will end up with so you can imagine this one through the Newman projection. round carbon, and in the back you have the living group of E and D. So this will come, it will attack here, will go in the bond and the living group will leave. So that will enter with this alkene. Okay. Similar thing for a more advanced one, you have this cyclohexane with promo CH3 and OCH3 minus. What are the chances of the product that you can have is this. Okay. This is your alpha, this is beta, this is also beta. Okay. If you abstract proton from here, you will get double bond this side. Each. We have both side two each here. So to understand which one will be the major product, in this case this is major, we have to draw the 3D conformation. To draw the 3D conformation, you have the bromo, and here you have the CH3, the other carbon you have proton. You can see CH3 is blocked the exact opposite side of the bromide. So only this proton is available. So definitely this will come and abstract this proton, and you will get the major product like this. Okay. And right choice of this is important to initiate the reaction. Okay, suppose for example you have a competition between the protons at two different sides. Okay, this is also beta, this is also beta. So when you are attacking with some base, it can give you two product. One is this one, and one is more substituted one. Yeah. So if you have a weak base, weak base, it will lead to this product to the more substituted one. You have a not actually weak. So if you have uh, non-bulky non-bulky base, and if you have a bulky base, bulky means there are much steric. Suppose for this one. much bulky groups on the base. So it will extract only this proton and it will get you a major product as the less stable one, less substituted one. So right choice of base is very important to get specific residual selectivity. So with that, we can complete this part and we can go towards some practice of questions. So this was practice set for this week. Which of the following forms more stable carbocation when it undergoes E1 reaction? E1 reaction, as we all know, what does it tell? That one carbocation intermediate, okay, should be stabilized. Now we check it. So if you lose this bromide for all the sides, first one will give you this carbocation, but a carbocation on a sp2 hybridized center, not possible. This one, if carbocation is generated here, it is a allylic carbocation, so it will have some resonance like this. And this will be stable, right? Similarly for the other two cases also, this carbocation not stable. On a benzene ring carbocation, forget about it. Okay, this is not stable at all. So this one will be the right answer. Next question, what is the major product of the following reaction? You have NaOCH3 and CH3OH. You can check this bromo and this methyl is opposite side. So surely, as we have seen here, that if they are in the opposite side, this part will be blocked from the methyl at the 180-degree dihedral side. Only with the other beta carbon, the proton will be available. So it has to be this side. Okay, here you have two hydrogen. One of them can be abstracted, so that we will get to have only one product, right? I can draw this one, the confirmation. So suppose we have bromo here, you have methyl this side, methyl. So you can see this part is blocked. Only this part you have two protons. So one of them can be abstracted and it will go here and bromine will be left. So next question says that typically how many transition states and intermediates are formed in the energy profile diagram of reaction undergoing E2. Okay, E2 means you have a rate that is substrate plus the base or nucleophile. And what is the energy profile diagram? This is reaction coordinate R and P. We have only one hump. So this is your transition state, not an intermediate. So one transition state, no intermediate. This is the right answer. Next question is choose the correct statement for the two anionic molecules. So what are the point of concern here? Nucleophilicity in water. Okay, basicity versus nucleophilicity in water. So what you know, water can undo each bonding. Each bonding will be much more for O minus than this minus, right? Because here electron is not uh, distributed more because sulfur is bigger in size than oxygen. Electron is confined here, so it can have much more hydrogen bonding, so reduces nuclear phylicity. Okay. So the scenario will be like O minus more basic, less nucleophilic, S minus less basic, more nucleophilic, because this is not that much stabilized or hydrogen bonded as compared to the oxygen. So let's check out the options. One is less basic and more nucleophilic. No, it is more basic. One is less basic. No, one is more basic and less nucleophilic than 2 and water. Yes, this will be the right option. For the next question, which of the following is the least reactive compound by SN2 reaction mechanism? For SN2, what you require? For SN2, substrate should be less steric, sterically hindered. Okay. So now compare in between these two here. You have one, two, three, much bulkier group here. Here you have the methyl group in the beta position. Here you have gamma position, and here you don't have. So this one will be, by far the most or the least reactive compound because there is only one proton out there. So one will be the most reactive, then will be the four, then two, then three. Three will be least reactive. So reactivity three is two, then four. Let us do the other way. Three, then four, then first one was eight. Okay. Then what is the nucleophile in the reaction shown below? Okay. So we can see here that is a second SN2 reaction. Okay, so water is acting as nucleophile as well as base, right? So nucleophile will be water. So in the first step, it will come and remove this one, OH2 plus. Then again water will come, but as a base, right? Okay. So next question is the reaction Q1, the first one. It is an SN2 reaction, right? So it should be backside attack of nucleophile. There is no chance of getting a stable carbocation here. Now, Assuming no other changes, what is the effect of doubling both the alkyl halide and the nucleophile concentrations in the above reaction? For SN2, you have R substrate plus nucleophile both. So if you double this, so what will be happen? 2x, uh, this is 2, this is 2. So overall rate will be R initial into 4, right? So it will be quadruple the rate, initial rate. Now, which of the following best describes the carbon-oxygen bond of an alcohol? Uh, uh, let's say simple alcohol will take is 3CH2 OH. Oxygen is delta negative, carbon is delta positive. So this is polar, delta positive at carbon, delta negative at oxygen. What is the major product of this following reaction? So H2O become okay. So let us just try to do it. H2 is very weak nucleophile, so it will go through what SN1 mechanism, right? So just try to do it. That will be your positive, and it will be 2 methyl group. But you have a chance to form a more stable carbocation. So there will be some rearrangement so that one methyl group will come here and you will get this carbocation. Okay. Now water can come and there will be one more proton abstraction stage so that you will end up with OH and this one is your major product. So why elimination is not taking place? Because this is very weak nucleophile. Okay, it will not try to take proton and then abstract proton and then you will end up with an elimination. SN2 will, SN1 will predominate. Okay, so this is the right answer. One. Which of the following characteristics are associated with the reaction in question 5? This is the E1 SN1 reaction. SN1 with rearrangement. So a nucleophilic attack from the backside of the C1 CI bond, no, this is wrong. Trigonal planar intermediate, yes, this is right. Uncharted mechanism with no intermediate, no, this is wrong. Inversion of configuration, yeah, that can be possible because in SN1 you have mixture and forms various stereoisomers, including a pair of enantiomer and pair of diastereomers. Okay, let us see. You have a rearrangement and you have this one. So in this case, if water is attacking, it can attack in just two sides. Okay, either from this side or from this side. So you will have two pair of enantiomers here, right? Not two pair, one pair of enantiomer, pair of enantiomers. Okay, so this various stereoisomer including one pair of diastereomer. So diastereomer possibility is not there, so this will not be right. So 2 and 4 only. Uh, just a second. Okay, this is not right. So only inversion is not possible. There will be retention also. In SN1, you will have both retention plus inversion. Okay, so this one is not right, but this can be right. So pair of enantiomers we know, pair of diastereomers we have to check. Yeah, you will have pair of diastereomers because uh, you are shifting from this methyl to this position, and here also you have one two possibilities of R S. Okay, so altogether there will be four, depending on this configuration of particular carbon center. So altogether there will be four. Okay, so this is right to 1 5. Next question is the major product of the following reaction is. So you have a base out there, and so in this case you draw it here, proton and Cl on the same side. So this is not a favorable conformation for the elimination. So it has to rotate. This bond has to rotate so that you will get to some another conformation where proton will be at the bottom. When it will be at the bottom, then this will be here, and on the top you will have the isobutyl group. And now you can insert the base. It will extract proton and leaving group will leave, so that you will have. Okay, so what is this isomer E or Z? Now you check it. This is your first priority group, and this is your first priority group. Okay, so these are on the opposite side. So this is E alkyl. Next is what is the right structure of the product of the reaction below. You have this base, and now similarly you draw the 3D conformation. You have Cl, right? You have this one, this proton, and this can be abstracted, and which will leave. So you will have this one as the answer. Next question is compound A reacts in E1 reaction with a weak base. Yes. What is the most likely structure of A? You have to get the starting material right here. E1 reaction, that means there is one carbocation. So from which carbocation it can generate? Generate from either here. Okay, most likely. So how this carbocation can be generated? So if you remove this one, you will end up at this one, and if you remove this one, you will end up at this position. Okay, now here you have a proton, and that can shift. so that we'll have a rearrangement to have the similar carbon because it is much more stable. So it will be 3 So this both will be right, 2 and 3. Last question is identify a polar aprotic solvent from the following: acetic acid, R, COOH. It is protic, Excel. These are non-polar, polar protic, ethanol, OH. It is also polar protic, acetone. Acetone is CH3, COCH3. Acetone is polar but non-protic. So this is the right answer. So we gained, very good information today about the substitution and elimination reactions, their energy profile diagram, and then we started, try to solve many of the questions, and I think it will help you. So with that, we'll close today's class and see you all on the next one. So thank you all.